You spent 3 years becoming a person, learning a face, a word, fear itself. But none of that is there. Not blurry, not faded at the edges, gone. Think about what you are missing. The first time you understood that the shape leaning over you was a face, and that the face belonged to someone. The first time a sound came out of your mouth and changed what a room did. The first time you were afraid, and the first time being held made the fear stop. That is the densest stretch of learning in a human life. You will never learn that fast again. And you have no access to any of it. We have a name for this: infantile amnesia. For most people, the wall sits somewhere around 3.5 years old. Before it, almost nothing survives. After it, the record starts. Test it on yourself right now. Go to the earliest thing you can find. Not the earliest story you have been told, the earliest thing you can actually see from the inside. Now look at what you are holding. It is probably a single frame. A patch of carpet, a doorway, somebody's leg at eye level. No before, no after, no sound. It arrives already cropped. And here is the uncomfortable part of that exercise. A good number of the memories people offer as their first are reconstructions, assembled later from a photograph on a wall or from a story an adult repeated until it grew pictures. You cannot always tell from the inside which of yours are which. The one you are looking at right now might have been given to you. That is the edge of the wall. Everything past it is not dim. It is absent. The obvious explanation is that a baby's brain is too unfinished to record anything. That there is no memory to lose because no memory was ever made. For about 100 years, that was the working answer. In March 2025, a team put babies inside a brain scanner and found out it was wrong. So where did those 3 years go? Start with what you were actually doing. At 2 months, you cannot hold your own head up. By 36 months, you are running, negotiating, lying badly, and using a grammar you were never taught. Nobody sat you down with a textbook. You reverse engineered an entire language out of overheard fragments, working from nothing but the sound of adults talking near you. You also learned physics, not the equations, the expectations. That objects fall, that they keep existing when they roll behind the couch, that a cup tips if you push its rim, and that the floor is where things end up. Every one of those is a rule you extracted from evidence, and you extracted hundreds of them by experiment, mostly by dropping things and watching. And you learned people, which is harder, that the same face means safe or dangerous depending on the shape of the mouth, that crying summons someone, that the summoning is not guaranteed, and that the delay tells you something. Before you had a single word for any of it, you were already running a model of other minds. None of that is stored as a story you can retell, but it is stored. You still speak the language. You still expect the cup to tip. Here's where it gets stranger. Your memory is not one thing. It is at least two systems running on different hardware. There is the one that holds skills and expectations, that lets you ride a bike without narrating it, that catches a glass before you have decided to catch it. And there is the one that holds episodes, a specific afternoon, a specific room, a specific thing that happened once and never again. The first system survived your infancy intact. Second, left nothing behind. That should bother you more than it does, because it means the machinery did not simply fail. Something took the episodes and left the skills. A total blackout would be easier to explain than a selective one. The classic answer points at the hippocampus, a curled structure buried in each side of your brain, the part most involved in binding an episode together. The where, the when, the what, stitched into one retrievable thing. In an infant, that structure is still under heavy construction, and one detail of that construction turns out to matter enormously. Your hippocampus makes new neurons, not only in the womb, throughout life, at a rate that is spectacular early and slows down after. In infancy, the region is flooded with them. New neurons sound like an unambiguous good. More cells, more capacity. In 2012, two researchers in Toronto, Sheena Jocelyn and Paul Franklin, proposed something less comfortable. What if the flood is the problem? Their reasoning runs like this. A memory is not a substance parked inside a cell. It is a pattern held across a specific set of connections between cells. Now, push thousands of new neurons into that circuit, each one wiring itself in, rearranging who connects to whom. The pattern does not get erased. It gets overwritten by renovation. They called it the neurogenic hypothesis. A brain building itself at full speed cannot hold a stable episodic record because the record is made of the very connections being rebuilt. It is an elegant idea. In 2014, they tested it, and the result was strange enough to publish in Science. They worked with infant mice, which show their own version of infantile amnesia. They form memories and lose them fast. The team suppressed hippocampal neurogenesis after a memory was formed. The forgetting slowed. Memories that should have vanished stayed. Then they ran it backwards. In adult mice, whose memories normally hold, they increased neurogenesis after a memory was formed. The adults started forgetting like infants. The effect ran in both directions. Slow the building and the record survives. Speed the building and the record degrades. And yet the strongest piece is the third one. Some species are born far more finished than mice. Guinea pigs and dagoos arrive with their eyes open, able to walk within hours, and most of their hippocampal neurons are already generated before birth. The team looked at infant guinea pigs and dagoos and found they did not show the same infantile forgetting. Then they artificially increased neurogenesis in those infants. The forgetting appeared. So the pattern holds across species, runs in both directions, and can be switched on in animals that do not normally have it. Now, the part where this has to slow down, because it is the part that gets misreported. Every one of those experiments was done in rodents. Mice, guinea pigs, dagoos. Nobody has manipulated neurogenesis in a human infant and watched what happens to their memories, and nobody is going to. The neurogenic hypothesis is a strong, well-tested account of forgetting in animals, and a reasonable account in us. The bridge to humans has not been walked. It has been pointed at, which leaves the original question standing. And in humans, a second explanation was running alongside it. Language. And episodic memory, the kind you can retell, is shaped like a story. It has a sequence, a location, a self at the center of it. Some researchers argue the capacity to hold that shape arrives with the capacity to speak it. That before you have narrative, you have no container to keep the episode in. If that is right, the wall should move depending on how much narrative a child grows up inside. And it does. In a cross-cultural study published in 2001, Chi Wang compared the earliest memories of American and Chinese participants and found the boundary is not fixed. Chinese participants reported earliest memories almost 6 months later than American participants. 6 months on a wall that sits around 3.5 years. The explanation offered is not that one group has better memory. It is about how much a household talks with small children about the child's own past. How much elaborate back and forth remembering happens out loud, and how much that talk centers the child as an individual with their own story. Where that talk is richer, the wall sits earlier. Your first memory has a date. That date was set in part by how the adults around you talked. But here's the assumption underneath every version of this. The neurons, the language, all of it, they share one belief. That the memories were never properly built. That there is nothing behind the wall. In March 2025, that belief broke. The problem had always been technical. To find out whether a baby is encoding a memory, you need to see inside a working infant brain. And functional MRI requires the subject to lie still and stay awake inside a very loud tube. Babies do not cooperate with that. So the question had stayed open, not because it was uninteresting, but because it was unaskable. A team at Yale, led by Tristan Yates and Nicholas Turk Brown, got it to work. Awake infants, aged 4 to 24 months, inside the scanner, looking at images while their hippocampus was recorded. Later, the same infants were shown those images again beside new ones, and the researchers tracked which ones they looked at longer, the standard way to ask a pre-verbal human whether something is familiar. Then they went back to the brain data with a single question. When an image was later remembered, had the hippocampus done something different at the moment it was first seen? It had. Higher activity in the posterior hippocampus while an image was being viewed predicted that the infant would recognize it later. The effect showed up clearly from around 12 months. Read that slowly. The encoding machinery was running in babies. The hippocampus was doing the thing it does in you right now and doing it well enough to predict what would still be there later. Which means the century-old assumption had it backwards. The memories were made. So the question changes shape entirely. It is no longer why your infant brain failed to record. It is why the recording became unreachable. The researchers themselves frame it as a shift from encoding failure toward retrieval failure. And that is a different order of loss. An encoding failure means there was never anything there. A retrieval failure means there is something there, and the road to it is gone. Be careful with that, because it is where this gets oversold. Nobody has recovered an infant memory in an adult. This is not a claim that your first birthday sits in a file waiting for the right key. It is narrower and stranger. At the moment those years were happening, they were being written down. What happened between then and now is the open question. And that is where the vertigo is. Think about what a retrieved memory actually is. Not a recording played back, a reconstruction, assembled now by the brain you have now, out of the connections you have now. Your infant memories were written into a hippocampus that no longer exists in that configuration. The neurons holding those patterns were joined by millions of others that rewired the whole neighborhood. The you doing the retrieving is built from different connections than the you that did the encoding. So the file is not corrupted. The reader is. And the two explanations stop competing at that point. The renovation that made you unable to reach those years is the same renovation that turned you into someone capable of reaching for anything. You did not lose those memories despite growing up. You lost them by growing up. Somebody lived those 3 years. Somebody learned that faces are people, that sounds change rooms, that being held stops fear. Somebody did the hardest cognitive work you will ever do and handed you everything they built. Your language, your expectations, your certainty that the cup will tip. And they kept nothing, no account, no version of the story where they are the one it happened to. You have photographs of them, you call them you, but the only honest thing you can say about the person in that picture is that they made you possible, and you cannot reach them. So go and look at that photograph again, the one where you were small enough to be held. You have always read it as a picture of yourself earlier. It is not. It is a picture of the only person who has ever done something for you that you can never repay and never remember. They built the machine you think with. Then the building of it erased them. Nothing is as ordinary as it looks.